Okay, so moving on, continuing with our plasma membrane, we're going to look at handout 3C, and that's just in our PowerPoint slide, just more talk about the plasma membrane, handout 3C and 3D. So sticking with that theme of our plasma membrane, because it is important, let's look at handout 3C. Handout 3C is still going to be mentioning ways that we can move stuff across the membrane. So we talked about a concentration gradient where we're going to go from high concentration to low concentration. And then we talked about an electrochemical gradient where we can move a charge, ions, across the membrane. That was handout 3B. Now let's look at some other ways that we can move material across the membrane. So handout 3C says transport across the plasma membrane. This is just processes to move substances across the cell membrane. It's how the cell metabolizes. So moving stuff in and out, we need to move nutrients in, we need to move waste products out, is essential to life. It says some substances can cross the lipid bilayer while others have to cross through channels. Those channel proteins that I mentioned back in handout 3A. Now, there are a whole list of things that the cell can do to move stuff across the membrane. There's two categories. We can move things passively, and that requires no energy at all. It's just physics that's moving stuff. Or we can move things actively through active transport, and if we do that, this requires energy to do this. Let's focus first on this passive transport that doesn't require any energy. Now, you may have heard of some of these terms. These are just ways that we can move a product through the membrane. So the first one is diffusion. And diffusion is just moving a product from high concentration through the membrane to low concentration. So small nonpolar molecules that we need to get in or out of the cell can move straight by diffusion from high concentration to low concentration. A lot of times it's like the plasma membrane's not even there. Osmosis is similar, but osmosis is going to move water from low solute concentration to high solute concentration. Now, this solute is usually a salt. So water is going to move from a low salt to a high salt. And our next handout in the next video, 3D, is going to talk more about osmosis. So I'll elaborate more on osmosis on the next video. We can also move things by pressure. So move things from high pressure to low pressure. That's called filtration. That's what our kidneys do. So our kidneys push fluid through them from high pressure to low pressure in this process, filtration. And then we've got facilitated diffusion. Facilitated diffusion is just like diffusion, where we're moving from high concentration to low, except these are larger molecules like glucose, for example, that need a channel protein. So it says it moves large molecules from high concentration to low concentration, but we need a channel protein, a door, to allow those molecules to move through. Now, these four things don't require any energy. The cell just does them due to the physics of molecules. We do sometimes need to pump molecules across the membrane, and this is where we would need active transport. So it says active transport uses ATP, that's energy, to pump substances across the membrane. So we burn energy to do this. A lot of metabolic processes require us to burn energy to move stuff across the membrane. And this is why we need to eat, because we need that energy to run our cells. So those are some examples. And as we get further into this chapter and other chapters, we will talk more about examples of passive and active transport. Now, there's another way we can move material across the membrane, and that is through vesicles. Vesicles are little pouches of plasma membrane that either bring in a product or excrete a product. So if we bring stuff in, that's called endocytosis. If things move out of the cell, that's called exocytosis. Now, there are two kinds of endocytosis. If we move large molecules through, we call it phagocytosis. And if we move small things through a vesicle, we call it penocytosis. An example of this is, let's say we have a immune cell. This is a white blood cell, and we have bacteria. We probably know that white blood cells eat bacteria. That's how we kill the bacteria. Well, in order for this to happen, we have to undergo phagocytosis. So a white blood cell is going to phagocytize or eat that bacteria. So imagine my little orange dots here are bacteria. The cell can engulf that bacteria with a plasma membrane and bring that into the cell to be digested. So what we form is this circle of plasma membrane that we call a vesicle. If we make a product, let's say we make a hormone, or we make some kind of product like sweat or milk or oil, something that a cell needs to make, a lot of times we make that product in the cell and we package it into a vesicle and then send that vesicle to the plasma membrane, where the vesicle merges with that plasma membrane and releases the product out. That process is called exocytosis. And this is commonly done when a cell makes a product like oil, sweat, milk, or a hormone or other enzyme. We release that through exocytosis. Okay, so this handout has just given us an idea of how we can further transport material back and forth in and out of the cell. So you should realize that the transport of material is really important. Now, in our next video, we're going to focus more on diffusion and osmosis, and I want to elaborate on those, kind of give you an idea of how exactly those two things are working. So next up is handout 3D, and we'll explore this plasma membrane just a little bit further. All right, I'll see you soon.